Okay, yeah, thank you, Mert. It's great to be here. Really exciting. My first time here at the Institute, and hope to have many interactions with many of you. And so I'm going to tell you what we're interested in from the perspective of biochemists. And we're particularly interested in stress responses here, and also how these stress responses are linked to diseases. And we think that for stress adaptation, actually, biomolecular condensates are very important. Now, just generally about stress responses. So there's two things you have to do when you want to, deal with stress. One of them is you have to detect the stress, right? There's different types of stresses: oxidative stress, heat stress, as you already heard from Alan. So you have to detect the stress, and you also have to then physiologically adapt to that. Yeah. So these are the two things that cells have to do. I would say that the phase behavior of proteins or macromolecules in general is very good for detecting, because the phase behavior is very much sensitive to things like, for example, pH or salt or all kinds of other physical chemical signals. And so by doing this, or by phase separating, you can have a switch-like response with which you respond to stresses and actually detect them. You can also, of course, when you form condensates, you have all kinds of different material properties, and of course, this can regulate macromolecules, and that is probably what's relevant also for the physiological adaptation. So the idea for us here is that phase behavior of macromolecules is probably integrally linked to stress responses, and I want to make a case today that this is actually true. And so I want to tell two stories, one of which actually picks up where Alan left off. So this is about heat stress granules in budding yeast, and the other one is stress granules in mammalian cells. So first of all, see these heat stress granules about which Alan also talked. And what you're seeing here is a protein that one tagged with GFP, and you can see that at 35 degrees, it's diffusely localized. But when there's a heat stress stimulus, then this protein assembles into these heat stress granules that Alan also mentioned before. And so we're trying to understand what these heat stress granules are, and actually also what their function is. Now, there's a lot of work that has already been done. Here, one thing I want to point out is that when you expose yeast to heat stress, one thing they do is they arrest their growth. This is yeast cell growth. They also stop general translation. They induce the heat shock response, and they produce chaperones to fight the heat shock. And this is, of course, the physiological adaptation. And this goes along with the formation of stress granules. And this has already been pointed out by Susan Lindquist here in 1986, where you can see, for example, here for Drosophila, this huge change. So this is basically loading the whole lysate of a cell before stress, which is C here, and then heat shock, of course. And you see there's a completely different pattern of proteins that are being produced under these two conditions. And of course, we have to explain how this pattern comes about. Now, we showed in 2020 that there's one thing that is actually happening here. So we did ribosome footprinting experiments, and we looked at the messenger RNAs and what these messenger RNAs have in common that are induced under heat stress conditions. And we found that heat shock mRNAs, they tend to have a less complex 5' UTR. So, mRNAs have a leader sequence, which is this 5' UTR, and it tends to be shorter, and it's also less structured. Whereas those mRNAs that encode housekeeping proteins, they tend to have 5' UTR, so they're more structured and also longer. And just keep that in mind, because I'm going to come back to this. So, of course, this raised the question for us, which translation factors are involved here in this? So I want to now introduce a few of these key translation factors that are required for general translation, but we think also for the stress adaptation. So one of them is this protein here. This is EF4G, which is a scaffold protein that binds to the 5' UTR. Then we have this protein here, 4E, which is a cap binding factor, and we have a helicase here that uses ATP to remodel RNA. This is EF4A. Together they form this EF4F complex. There's other factors. Alan pointed out this one. This is the stress granule protein, but also one that binds to the poly-A tail. And there's another helicase here, DAT1. Importantly, these factors are required for active translation of messenger RNA. So very often you see the formation of such circles here, and this is required for active translation of messenger RNAs. Now, so these are the key factors, and then we looked at them first before stress, and then, of course, during stress. So you see, this is at 30 degrees. You see that these factors are diffusely localized. But then when we stress the cell, you see that these two guys here, 4G and 4E, they assemble into heat stress granules, and they overlap with PAP1, a stress granule marker. But 4A does not. Yeah? And this is important, because I'm going to come back to this later on. So G and E form stress granules, A doesn't form stress granules. And they also dissolve after an hour at normal temperatures. Now, being biochemists, we then wanted to reconstitute these structures. And so we purified all these factors. We made messenger RNA. And so with mRNA and this EIF4G protein alone, you get condensates in a heat-dependent manner. I mean, there's a lot of other data that I'm not going to show you here. But importantly, so you have these 4G mRNA condensates that form in a heat shock-dependent manner, and then they recruit this 4E binding protein, this cap binding protein, as a client. They also recruit PAP1, but they do not recruit 4A. And remember, this is exactly what we had seen in the cells. 4A stayed diffuse, but E and PAP and G, they formed punctile. So this suggests that we can actually reconstitute this here with this complex mixture of components. But the question then is, why is ER4A not recruited into these condensates? For this, we did a binding experiment here. This is an anisotropy binding assay. We're titrating the amount of EIF4G, and we have different binding partners, RNA, 4E, and 4A. And you can see that initially they form a complex, yeah? But now we heat up the sample, and then you can see that 4A is no longer able to bind. Yeah, you see the difference here. This is the binding curve, and this is no longer associating now with the complex. And to cut a long story short, so there is a change in this 4A binding domain here that dissociates 4A, and we think that there's a heat sensor domain in this region here, and that undergoes a conformational change and then releases essentially 4A, which can no longer bind because there's a heat-dependent conformational change here. So then, and this actually goes back also to what Alan pointed out, so the question really is, so there are condensates clearly forming, but the question is also, are there smaller things that are also important that make up these condensates? So what we did here is we formed these MRMPs that Alan mentioned, and they have a distinctive size. So this is DLS, so this is the cumulant radius. So importantly, when we now heat up the sample, what I have to say is that this is below the saturation concentration. So when we heat up the sample, the size remains the same. But what was really interesting is now if we actually do this above CSAT, so we form the condensates, and then we dissociate them with salt, we get the same sized structures. So we again get 50 nanometer particles. And this is suggesting that these condensates that form, that they're composed of these smaller mRNPs, and this is actually what they are made of, yeah? And the great thing is we can now reconstitute this here using reconstitution biochemistry. But of course, the question then arises, what is the functional consequence of this? And for this, we did in vitro translation assays. So you break open a yeast cell, you make an extract, and then you program it with a messenger RNA, a reporter messenger RNA, and then you can read out the luminescence, and this tells you, of course, about how much translation this lysate can do. So we did this here using a reporter mRNA. We're titrating our factors, so we have these purified factors, and you can see that with increasing amounts of this EF complex, but also including PAP1, we get a several hundredfold induction of translation. So our system clearly is working, so we can read out translation. Now then, of course, we wanted to see, so what about these condensates? Now we can reconstitute these condensates. Do they promote translation or do they repress translation? So this is an increasing temperature here, and then we're measuring here the fold change in translation. And of course, we looked at this in the microscope, and you can see about here the formation of condensates. And so the formation of condensates very nicely correlates with repression of translation. So these are the condensates. But I showed you before that we can also make these MRNPs. So what about these MRNPs? So interestingly, before heat shock, the MRNPs are as competent in translation as the mRNA only. But then when we heat stress the mRNPs, then there is a significant loss in translation activity. This is suggesting that already the mRNPs are translationally inactive, but they assemble then into this bigger condensate, which may have additional function here, but we don't know that yet. So the important unit seems to be the mRNP here that probably undergoes a conformational change, and then it's inactive in translation. So I showed you this before, this switch in translation where, we had heat shock messages and we had housekeeping messages, and there was a difference in the structure of the 5' UTR. So can this now explain the switch in translation during heat shock that we saw? For this we again made an extract, but here now we're using yeast cells that are already heat stressed. And importantly, there is a centrifugation step here. So that, of course, means that translation factors that are assembled into heat-stressed granules, they will be depleted from the lysate. Yeah? And so now we're using this heat shock lysate to see what the activity of these different messenger RNAs is. And we have two types of messenger RNAs here. We have those encoding for housekeeping proteins and those encoding for chaperones. And remember, they have differences in the complexity of the 5' UTR. And you can see that the housekeeping mRNAs, they can barely be read when you now titrate this soluble factor 4A, which is required for the remodeling of the mRNA during translation initiation. So the housekeeping ones, they cannot be efficiently translated, but the stress mRNAs, they can be translated. Yeah. So this suggests that here heat shock mRNAs are now preferentially translated, and that is because of the differently structured 5' UTRs, and also because there's just this one factor here available to them. Many factors have been assembled into heat stress granules and they're gone, essentially, because of the centrifugation step. Now we wanted to test this, so we titrated back in the factors that we know assemble into heat stress granules that are probably missing. And you can see when we now increase the concentration of GE, PAP, but also DET1, another factor, we can bring the housekeeping translation back to high levels. And so this, suggests really that there are factors being depleted into heat stress granules during heat shock, and this leads to a differential treatment of the mRNAs because only the heat mRNAs can be translated by just 4A alone. And just to summarize this, you have all these factors here involved in promoting mRNA translation in active regrowing cells, but then when heat shock happens, there is a sequestration of these old mRNAs in these heat stress granules. But now there's, of course, newly made mRNAs. Alan also pointed that out. There's newly made mRNAs. Those have these simple 5' UTRs, and there are now some factors remaining, such as 4A. This still soluble, and that is sufficient now to promote the translation of these more primitive 5' UTR containing heat shock messages. And so I think this is a very, very nice explanation in the model for how translation is regulated during heat shock. A very last thing is also, of course, and Alan pointed that out, so that these heat stress granules have solid-like properties, so they're very different actually from mammalian stress granules. And when they dissolve, as shown here, chaperones are actually required, so molecular chaperones. So this has been shown by multiple groups, and we just recently also reconstituted this step where we added a whole bunch of factors HSP40, like this one here, HSP104, HSP70, and then, of course, this is looking at this fact that that one, which is a helicase, and the assay that we used is, so this is the unwinding of a double-strand RNA, which leads to a change in Fred signal, and only when we have the combination of all these chaperones, we can disassemble these condensates. And so this suggests also that there are conformational changes induced by the heat stress that lead to the formation of these condensates and MRMPs, and they need to be reversed by chaperones in order to make these proteins active again. All right, now I want to switch to the second part, which is stress granules in mammalian cells, which, as we already heard, they have very different properties. They're more liquid-like. So this is, what you're seeing here is a HeLa cell. The black thing here in the middle, this is the nucleus. And this HeLa cell is expressing a GFP fusion to an RNA binding protein, G3BP. And we're stressing the cell with arsenite at time point 0. And you can then see how these condensates form here and how they fuse. And so this protein G3BP is actually key for this process. So, but first of all, I want to tell you a little bit about mammalian stress granules and what is known about them. So what happens in mammalian cells during stress is that they release the messenger RNA from polysomes, and that messenger RNA then assembles with RNA binding proteins such as the G3BP protein and also helicases and some signaling molecules into these stress granules, and this has been linked to translation inhibition and signaling, although most of the data is correlative. We focused on this G3BP protein because genetically, it's also work from Paul Taylor, has shown that this is genetically absolutely required. So this protein, in the absence of RNA, doesn't form any condensates, but in the presence, it forms very nice condensates. And these condensates are very dynamic, so they show these fusion events here, and they also recover very quickly within seconds when you photobleach these condensates. Now, we think that the association of G3BP with RNA is the first step of stress granule formation, but then many other factors are coming in, and we tested this here also biochemically. So you initially form these G3BP RNA condensates, and then there's a whole bunch of other RNA binding proteins that are then recruited downstream to form mature stress granules then that have the composition of also, or is approximating the composition of stress granules in cells. This is what we did a couple of years ago, but then we asked the question, what about RNA? Because at this point we mostly focused on the proteins. So this is new work that we published this year in Molecular Cell, where we focused on the RNA, and one of the experiments that we did together with Michael Schlerf here is that we used an RNA hairpin with 2 dyes on it, and we formed these G3BP RNA condensates, and to our surprise, we saw a big difference in the signal that we got from the hairpin when we compared inside and outside the condensate. So outside the condensates, we have a strong Fred signal, a strong Fred efficiency. But inside, the Fred efficiency goes down dramatically, which is suggesting that the hairpin is opened up inside the condensate, which was actually a big surprise to us. So we think what's going on here is that, of course, with a certain rate, the hairpin will unfold, and then this G3BP protein has a very high affinity for this single-stranded RNA, so it binds to it with a very low KD, and then associates with it and stabilizes it in the open conformation. That, of course, suggests now that you have many mRNAs inside condensates that are single-stranded and unstructured, and that could, of course, promote the formation of structures. So there could be base pairing between RNAs, there could also be these kinds of entanglements or loops happening, and so this is then what we wanted to test next. So does G3BP promote intermolecular RNA-RNA interactions? So we did a whole bunch of experiments. I'm just going to show you a few, one of which is the mRNA is barely dynamic. You can see this here in photobleaching experiments, while the protein component is super dynamic. So this is consistent with this idea that the RNAs are interacting with one another. We compared the stability of these reconstituted condensates made of mRNA or poly-A RNA. Poly-A, of course, cannot base pair, but mRNA can base pair, and you can see that those made with mRNA are more stable, suggesting that there are base pairing interactions. We also then looked at helicases. GDX3X is one of them, and this is here the mobility of the mRNA in the absence of the helicase, and then in the presence of the helicase, the mRNA is much more mobile, of course, because it remodels these base pairing or double-stranded regions. And actually, another very nice movie here is when we form structures with RNA in the absence of a helicase, but then we're adding the helicase, we can see this transition here from a solid-like assembly to a liquid-like assembly. So clearly the helicases are remodeling also the material properties of these granules. So RNA-RNA interactions are very important for the stability and material properties of these condensates. But what is the function of all this? So we turn to our in vitro translation assay again here. So we're comparing now, this is again a reporter mRNA. We're in this case adding just the mRNA. This is how much translation we get. Now we're adding G3BP, we're repressing the translation. So this is suggesting that sequestration of the mRNA inside the condensate is repressing translation. Now we have a mutation here, the delta NTF. That one is important because it prevents dimerization and condensation of G3BP, but it doesn't change the affinity for RNA. But then you see this. So that should suggest that it's the condensation inside the sequestration of the mRNA and not the RNA binding that represses the translation. And then we're adding also the helicase now, and we can increase, of course, the dynamics as I showed you before, but we also increase the translatability of these messages. Same is happening in cells, where you see that cells that have condensates have a lower translation level. So to summarize this, we think these G3BP stress granules in mammalian cells, that they are forming together with RNA, and they promote RNA-RNA interactions, and that helicases are remodeling these RNA-RNA interactions, and that this is required to make these RNAs translatable again. All right, very last 5 minutes. I want to switch now to disease. It's a story that we just published with Tony Hyman 2 weeks ago. So there's this RNA binding protein, TDP43, which is known to form these aggregates here in the cytosol. It's a nuclear protein that aggregates in the cytosol. The reason why we were interested in this protein is it is also localizing to stress granules. And there has been this idea that stress granules could be a precursor of these pathological aggregates. But this is controversial, and I want to show you some data that indeed suggests that it could be that stress granules are a precursor for at least these TDP43 aggregates that we find in patients. Now, I showed you we can reconstitute these minimal stress granules with purified components. We also did this here. So this is G3BP. TDP43 is nicely sequestered inside, and that's actually because of the RNA. And then we looked at this over time, and the important thing is focus on this TDP43 channel here. So initially, the TDP43 was diffuse, but then you saw this demixing, as we call it, intracondensate demixing happening on the surface, it seems. Here, you see this again. So it seems that TDP43 is unstable. It forms its own phase, which also excludes G3BP, and we also showed RNA is also excluded. So somehow the protein isn't happy inside this condensate. We then wanted to see what is this actually. So what are these demixed states of TDP43? We found that the demixed TDP43 is barely dynamic, which would be consistent with that the protein is forming aggregates here. Then we purified a chaperone, a small heat protein, which specifically recognizes misfolded proteins. And you can see that it nicely co-localizes with the TDP43 here, suggesting indeed we're dealing with misfolded TDP43. And so that is, of course, reconstitution. So I showed you so far we can we can rebuild these stress granules, we can observe an interesting phenomenon. But of course, it doesn't mean at all that it's relevant in a cell, right? So what about cells? So what you're seeing here is, this is one stress granule, and it's really blown up, so it's a big stress granule. And so here you see, in green, this is TDP-43, and in red, this is our G3BP stress granule marker. And we're adding arsenide, which is also oxidative stress at time point 0. I'm just going to run the movie now that you can see what happens. So that's pretty cool, right? Because it shows exactly the same behavior as in our minimal reconstitution system, right? Which really gives us confidence that we're reconstituting the real deal here. So indeed, TDP-43 is somehow unhappy inside these stress granules. And of course, we then wanted to figure out what is destabilizing TDP-43. And we had some hints that it could be oxidative stress. And so you can see this here. So this is the demixing happening in vitro, but then we can completely block this with a TSAP, so which is a reducing agent, suggesting there's oxidative stress, and you also, of course, have oxidative stress in cells because of the arsenide that we added to stress the cells. But it seems like even the background oxidation level is sufficient here to induce the demixing of TDP-43. But then we asked, so what is the molecular basis of all this? We teamed up with Jitay Mittal, who found that there's one domain in TDP-43 that is folded when it's bound to RNA, but when it's unbound, two cysteines get exposed, and this is consistent with this effect of oxidation that we had seen. So the idea that we had is that this promotes the formation of disulfide bonds, and this is actually why you see the demixing happening there. And we tested this then in cells, so we went back into cells. This is just the phenotype that we saw before, the demixing phenotype. Now we can mutate these cysteines, and you can see with another mutation combined, we can completely block the demixing process now in cells, suggesting that we really have a good molecular understanding now of of this intracondensate demixing process, that we can block it in cells. To summarize this, so TDP-43 is accumulating in stress granules. It's unhappy there under oxidative stress conditions, and we can talk more about why we think that is. And then it forms aggregates, or it misfolds and forms these aggregates. It looks like at the interface, and this is driven by changes in especially in the RNA recognition motif, which exposes cysteines, which then get oxidized to disulfide bonds. And of course, the C-terminal cross-beta sheet is also forming, but I'm not, I didn't talk about this much, but you can ask me about that later. And with this, I would like to thank a lot of people involved here, and I'm happy to take questions now.
Really nice talk, thanks. So in the movie of the stress granule in the cell, there was a vacuole, and there was demixing on the surface of the vacuole as well as the surface of the outside, and that was different from in vitro. Yeah. So do you often see vacuoles in stress granules? And
so what this vacuole is, I don't know. Roy Parker said that he saw that there's these different territories, and that G3BP has its own territory, but there are other factors of stress granules which are occupying other areas of stress granules. What these vacuoles really are, it's just there is no G3BP there. There's probably some other RNA binding proteins there, and somehow it happens on the surface there. It's an interesting observation, but I really don't know what's going on. So
that interface is presumably different than the outer interface.
It's true. That's true, and probably, yeah, but yeah.
Very cool. Thanks.
Yeah.
Beautiful talk. For your experiments, when you look at RNA and RNA-RNA interactions, so you look at poly A versus mRNA, can you set up experiments where the mRNA base pairing propensity changes, and does that then map onto the changes in material properties?
So you're asking whether we use different types of mRNAs?
Yeah.
So we haven't done this systematically, but it would be nice to test different RNAs for which we know that they would interact more, right? So that's the next step. And of course, there are also RNAs that have repeats. I mean, this has been known. Ankur Jain showed this, that RNA can phase separate on its own, and they have very often these repeat regions. And they would be more interactive. So this would all be very interesting to see how this scales with the stability also. But we haven't done that yet. Yeah.
Simon, the behavior for TDP-43 that you see is really remarkable. So I was wondering, is it really the change in redox potential upon oxidative stress that is key, or is it the high local concentration that would be enough even under regular oxidative conditions in the cell?
Yeah. I mean, several things have to come together here. It is the high local concentration, yes, that is very important. It also is the fact that it's unbound from RNA, so we think that the RNA that is in stress granules is not the right one. And so therefore you have very dynamically binding TDP-43 at high concentration locally. And so this is important. Then the oxidative stress has to be present, otherwise you wouldn't be able to form the disulfide bond. So there's multiple things conspiring here to give you this outcome. The reason why we think that these aggregates here are a problem is, and this maybe goes also to your question, I mean, people see also TDP-43 aggregating outside of stress granules. So there are smaller things forming, but they're so small, it looks like in our data right now, they're small enough that they can actually be dealt with. Yeah, so that there's autophagy and there's protein quality control, they can get rid of them. But these bigger things that form in stress granules, they hang in the cells for weeks sometimes, you know, they can't get rid of them. And we think it has to do with the fact that they're too big for autophagy to engulf them and get rid of them. And it's also because of the cysteine bonds. And so there's something about the size of these things that, you know, is the problem for the cells, we think right now.
So I think the lady that was sitting down was first, and then Tanya.
So I had very nice presentation. So one question that I had, that when it is like demixing around the sides, did you try to do like, because it looks like that there is a change in the material property across the condensates itself. So did you like do any lifetime analysis or
so which condensate?
Like where the TDP-43 is like demixing within the stress granule towards the sides. So does it looks like that towards the sides, if you like towards the side, if you like do photo bleaching or something, it probably won't recover, but the rest of it remains liquid. Does it?
So we did this photo bleaching experiment where we bleached the mixed and demixed area, and the mixed area is still very dynamic of TDP-43, and the demixed region, when we photo bleach that one, that progressively becomes less and less dynamic. So suggesting that there's more and more of these disulfide bonds forming, and so there's a more and more cross-linked aggregate that forms with time. And so we can observe that over multiple hours. So there's a progression here from a more dynamic, initial dynamic state, and you can also see fusions, for example. So this is initially dynamic, but then it more and more becomes arrested, and that's probably because of more and more cross-links forming.
Yeah, because I think it is also in the lines of the study that for maturation of condensates, the solidification kind of starts around like the edge, and then it like moves inwards. So this is also kind of in lines with that it is first sequestering towards the edge, and then it is like...
Yeah, so clearly the interface has an important role here, because it looks like it's always happening at the interface. It could be the orientation of the molecules. We don't know. I mean, this is something that we have to work out, how the interface, in what way it contributes to this process. Yeah, and it could also be, of course, that material comes from the outside, so this is not so, it's difficult to disentangle that, yeah.
Thank you so much.
So that was actually exactly what I wanted to ask about, whether you know whether material comes from the dilute phase, and then because you have oxidation processes, you get this
that could well be. That could well be, and I think it is initiated probably there. I mean, this is a very difficult. I mean, you just had, you know, H&RMP. You did something similar, and so you actually see that it's coming from the dilute phase. At least the material is coming from the dilute phase. And it's very likely to be the case, yes, but we haven't done, I mean, you have done very systematic experiments. We haven't done that yet.
Of course, on a different system, right? It could be different in your system, which is why I'm so
It's also, I mean, I should point out these aggregates that we're looking at here, I think they're quite disordered. They're not amyloid-like yet. They may be at the end, And so that is also maybe a difference to your system, that we have less ordered aggregates that we're looking at here.
We have a person here.
Hi. Oh, okay, there.
So the cysteines are really interesting. So potentially you could, like that's like a main mechanism for TDP disease progression. Do you know if that's like conserved evolutionarily, or if you can maybe cap the cysteines and prevent disease progression that way?
Could you repeat the question? So,
like those two cysteines, like they need to form a disulfide bonds.
Yeah, so that's what we think this is. I mean, we haven't done systematic mass spec experiments to really confirm this, right? But at least genetically, we made all kinds of mutations and combinations that suggest that there's actually intermolecular disulfide. We still have to do the molecular analysis of this.
Okay.
Yeah.
Yeah, I think a gene therapy.
Because, I mean, it could also be that it's intramolecular, but it looks like because of the change in material properties and so on that it's mostly intermolecular, and also because of the high concentration and so on and so forth. Yeah.
So we have a comment on this mic, on the left mic.
Really exciting. What is the fate of these stress granules after you have those TDP condensate? Are they, in the end, getting out of there, staying? Do you still see movement inside those stress granules over time? So
I mean, what happens eventually with
eventually with those, yeah.
So when you turn off the stress, the G3BP RNA will dissolve, and
still those clusters are.
Yeah. So what we think. I mean, the way we think about stress granules in mammalian cells is that there's an equilibrium here between ribosomes and sequestration and translation. And so mRNAs, whenever they're available, they can be also translated again. And so when you go back to recovery temperature, or in this case, no oxidative stress, then the stress gets smaller and smaller because the mRNAs are basically migrating back to the ribosomes. But the TDP-43 remains behind. So that is not dissolving. So the G3B is all gone, and you still have these aggregates floating around. We have some nice movies I can show you later. And those guys hang around for a very long time. And I think that is the problem. Yeah.
As more solid, like. Yeah, I get it.
Yeah.
Okay.
Nice talk. I have kind of two related questions. One, how many proteins have these kind of RRMs that have dicysteines? And do you think this is a general mechanism to start thinking about how folding domains might destabilize in the context of a distinct environment? So like, instead of thinking about ground state structures and things like this, we really ought to be thinking about the folded domain stability as something that...
Yes. I think that is something that is actually very important here. So, I mean, this RM that in the TDP-43 case is something that is quite unstable when the RNA is off. So, and every time it's off, it's like a globule state almost, like, and then it exposes its interior, and so that is a problem. So I think very often when RNA is unbound from these RNA binding proteins, they can cause a lot of trouble. But also for the heat stress story, I mean, it's a similar, so there's this 4A binding domain which undergoes heat-dependent conformational change, which is very small, it seems, so we don't see much of a, change in tertiary structure. But it seems that stresses lead to changes in conformational states of proteins, and then that has all kinds of consequences for disease, but also stress responses. So in the case of this 4G story, the yeast story, I think it's a programmed response that the cell controls, and there's evolution that has shaped these conformational states. In the case of TDP-43, there's a disease situation where multiple things have to come together, which then leads to the unfolding of this domain, and then the whole process gets going. Yeah.
Yeah, I'm trying to get my head around sort of the conformational transitions and where the cysteine disulfide comes in. So you're in high concentration, you've deprived the RNA, so you have this molten globule-like state. And then is the thought that the disulfide linkages are sort of locking you in one material state that aggregates, or you aggregate and then you form the disulfides? Or so where does it fit into the conformational transitions?
The demixing process, I mean, because we can completely block it with a reducing agent, suggests that the demixing process and the formation of the disulfide bridges, that this is basically linked, right? So the formation of these bonds is triggering the demixing, and then also you see this progression over time with getting more and more solid-like, suggesting there's more cross-links. Of course, we have to do the molecular work to link all these cross-links now to changes in material properties, and you know, that we haven't done that yet. It's also not going to be easy, but that'd be, of course, very nice to have, yeah.
Okay, so I think we have, okay, Tanya, I think, has a question, and then David, and then the person behind, what's your name? Yeah.
I'm trying to put how you talked about the translation initiation together with Alan's talk.
Yeah.
And you know, for the non-experts, right? So you're showing the mRNA as a circle that is held together by these initiation factors and PAB1, which would exactly fit into the picture of the ends being important, right? Yeah. So the idea is then that this specific complex that is forming, that only that can undergo some sort of conformational change that would trigger condensation?
That's the idea. I mean, it looks like, I mean, at least for G factor, and I mean, Alan showed it for PAB, there's also DET1. All these factors undergo a conformational change as a response to increase in temperature. And you also all find them in heat stress granules, and they also become insoluble. I mean, Alan showed that. So there's some factors that are sitting there, and they're probably coordinating this whole process. And what the key factors are, that is still completely unclear. Whether they are all required, whether there's just a few factors required. I mean, this we have to work out. Yeah, it's just, I mean, we know for 4G, for PAD, for DET, we know that they do this, but whether they are key, that has to be shown in the future, I guess. But Alan, maybe you want to...
Sorry, we have a queue, so David, and if there's time, Agnes.
Yeah, thank you. I have a question to the first part. And if I understood you correctly, you showed that translation is already suppressed in these RNPs by itself.
Yeah.
So I have a slightly provocative question, whether you actually need condensation at all, or would it just work?
Maybe not. Maybe not. I don't know. I mean, at least it looks like the mRNPs are already repressed. But it could be, of course, that there's some additional function here, right? I mean, you can imagine that they may be taken up by autophagy or these kinds of, I mean, who knows, right? But we haven't looked for this. I guess, would you agree with that, Alan? I mean, that it looks like that the big structures may not be absolutely required here, right?
Oh, yeah. For
okay.
Most of the biology.
Okay. Yeah, for the biology, at least. Yeah.
Yeah, so my question is, what do you think is causing this time delay in the formation of the TDP43 condensates on top? And is it the same in vitro and in vivo, like the same time scale?
So,
I mean, for a while they are demixed, for a while they are completely mixed, right? And then there's a demixing. So there is a time delay. So something is changing.
So that it takes a while for this to...
Yes, it takes quite a while.
Yeah, that's a good point. I don't know. I really don't know why this takes about an hour in this case.
Because I thought there may be other factors, but you see it in vitro as well, just with two components.
Yeah, so in vitro it also takes quite a while, right?
But is it faster in vivo?
And, I mean, we do know that I mean, at least I can speak about in vivo. We do know that chaperones, when we titrate them in, that they can block this. So definitely there will be factors that can block the misfolding and aggregation. But of course, we don't have these in vitro. So yeah, good question. I really don't know. Yeah.
Yeah, I just wanted to talk about what Tanya and Simon talked about. I think the best way to look at what's happening here is that we're making a bunch of observations, and there is no complete picture just yet. I mean, one of the exciting things about being in the field is that if you had looked at these questions, 5 years ago, we would have had a pretty clear idea of what's happening, and now it's very clear that is not what's happening, and there are all sorts of other things that are happening. So Simon's demonstration that these individual factors autonomously sense temperature and do things, right? Like, we're not exactly sure of what is happening in the cellular context, but it is totally clear that that is involved in some way in regulating translation. One of our big questions is how much of the RNA is actually coming with that stuff versus those things are condensing. and then separately being recruited to stress granules. That's the behavior that we see with poly A binding protein, to our great surprise, right? Like it lets go of RNA, condenses, and then gets dragged to stress granules where all the RNA is, so it's not at all what we would have expected. So part of the fun here is to get at, like, how are the proteins actually sensing these different conditions, temperature and pH, back to Ali's talk, and then what are all the moving pieces that the cells seem to be using to accomplish their actual biological ends. And I think one of the fun bits is, what if the cells actually know what they're doing, right? And a lot of this is purposeful, not the TDP-43 stuff, right? But that earlier story, the cells have sort of planned this evolutionarily speaking. We'd have to figure out what they're doing.
So let's thank Simon again.